DNA transcription is essential to life. It is the first step of many in the production of cellular proteins. A cell uses different regulatory mechanisms to vary the rate of gene transcription. It is the balance of these mechanisms that controls transcription and ensures cellular survival. Transcription is the process of creating a single strand of RNA from a double-stranded DNA template. Initiation of transcription requires several tartar box-associated factors, the most important being tartar box binding protein, or TBP. Tartar box-associated factors in TBP make up a complex called TF2D. TBP binds to the tartar box, inducing a 70-degree wedge in the DNA to make it easier for the enzyme RNA polymerase II to bind to the promoter. Activator proteins also bind to specific sites along the DNA. Multiprotein complex mediator then interacts with the activator protein, RNA polymerase II, and the transcription factors to complete the transcription initiation complex. RNA polymerase is released when transcription factor TF2H phosphorylates RNA polymerase, initiating transcription. Not all regulatory processes are controlled by transcription factors. Epigenetic processes refer to genetic modifications that affect gene regulation by changing the DNA conformation. Within the chromosome, DNA is tightly packaged into chromatin. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of two helical turns of DNA, wrapped around a core of 8 histones. The histones are involved in maintaining the nucleosome's shape and structure. To allow transcription to occur, histones are acetylated by histone acetyl transferases. This unwinds the chromatin in a conformation known as euchromatin. This makes it more accessible to the transcription complex. Histone deacetylase deacetylates histones, causing chromatin to wrap up tightly in a conformation known as heterochromatin. Genomic imprinting is an example of epigenetic inheritance. Epigenetic inheritance refers to a heritable change that does not occur as a result of changes to the DNA sequence. In genomic imprinting, the paternal or maternal gene copy is specifically expressed over the other. Perhaps the best example of genomic imprinting is imprinted gene insulin-like growth factor 2. Expression of both copies can lead to cancer. The mechanism is as follows. Insulators are genetic elements that block the effect of enhancers on the promoter. In genomic imprinting of the IGF2 gene, CTCF protein binds to the maternal gene's insulator element, blocking the effect of the enhancer, silencing the maternal gene. In the paternal gene, the insulator element has been methylated. This prevents CTCF protein from binding, so the enhancer activates IGF2 transcription. RNA interference is the process whereby RNA molecules inhibit gene expression by neutralizing specific messenger RNA. MicroRNAs are derived from actual genes and begin as primary transcripts of pri-microRNA and are usually transcribed by RNA polymerase II in the nucleus. The pri-microRNA transcript falls into a stem loop structure. Then, DROSHA, a nuclear RNase enzyme, interacts with the specialized RNA binding protein DGCR8 to form the microprocessor complex. The complex cleaves the pri-microRNA transcript, producing what is known as the microRNA precursor. Pre-mRNA is then transported into the cytosol through exportin 5 for further processing. In the cytosol, the precursor is cleaved by dicer endoribonuclease. To produce a 21 to 24 base pair molecule of microRNA. The microRNA consists of a passenger and guide strand. MicroRNA then associates with the Argonaute protein that is dedicated to microRNA-mediated silencing reactions. The passenger strand is then released, and the resulting structure is called the RNA-induced silencing complex, or RISC. Gene silencing can then follow one of two routes. In plants, RISC can target specific messenger RNA strands thanks to the remaining guide strand and directly cleave messenger RNA with help from associated factors and ATP. In animals, less extensive matches between the guide strand and messenger RNA means RISC prevents gene expression through inhibition of translation. Both processes result in eventual degradation of the messenger RNA. There are two main types of hormone: those that bind to external receptors and exert their effect indirectly using secondary messengers, or hormones that can enter the nucleus directly and affect transcription through binding to DNA. Steroid hormones help control metabolism and immune function, amongst other things. They are able to enter the nucleus and affect transcription regulation by binding to DNA directly. A major class of steroid hormones are glucocorticoids. The principal glucocorticoid hormone is cortisol. When cortisol enters the cell by diffusion, the hormone then binds the glucocorticoid receptor in the cytosol. This causes heat shock protein 90 to dissociate, revealing a nuclear localization signal, which was masked by heat shock protein 90. Then, the glucocorticoid receptor binds with an additional glucocorticoid receptor hormone complex to form a dimer. This then migrates into the nucleus. In the nucleus, the receptor binds the glucocorticoid response element, activating transcription of genes associated with metabolism, immune, and homeostatic functions.